we're going to start with our potential energy source, if you think about it in GChem terms, the potential energy. And that is carbohydrates, lipids, and proteins. These are all the things that we eat, okay? And that we think traditionally have caloric value. We talk about that in nutrition as being calories. Calories is just a measurement of energy in most of the classes that you've learned, either GChem or organic. We call about kilojoules. And if you actually go to Europe or travel overseas and you pick up a Snickers bar, instead of calories, they actually have kilojoules in terms of energy. And so it's just an energy value that's associated. Also, there's nucleic acids in the mix, okay? You actually do ingest nucleic acids with your proteins, but they're in such small quantities that we don't think about them as a caloric intake in terms of what we eat, although we have to degrade them. The nucleotides that we talk about here are what we degrade internally more so, and they need to be thought about as we go through the process. And so potential energies. When you eat them, you have a large source of energy in one form. Now, eventually, we're going to do a simple combustion, okay? Do you remember that reaction in general chemistry, combustion of a hydrocarbon, right? What is the products that you're supposed to predict from a combustion of a hydrocarbon? CO2 and water, that's it. And when you say combustion, that's a reaction of a hydrocarbon plus what? What's the other reactant? Oxygen. Combustion is oxygen, is burning, okay? And in most of the cases that we experience, okay, we are also going to use oxygen, and we're going to produce water, and we're going to produce CO2. So we are essentially burning the hydrocarbon inside our bodies effectively. Now, inside the chemistry lab, if you burn something, there's fire, and the energy is released as heat as the majority. We're going to do the same net reaction, but through a series of different reactions, so that instead of the energy all converting to heat, which is important for us to keep our body temperatures, but is not the majority. We want to extract that energy in a different way, even ending up in the same places. So we have to do, instead of one reaction, we have to do hundreds of reactions to convert those energy molecules into something that is usable, okay? Now, we are going to use lots and lots of pathways to do that, and really, this is a very simplified pathway scheme for what actually occurs. And so you see arrows, it's almost like a roadmap of what goes on inside the body. And this roadmap, just like a lot of our roads, can go both ways. There are some one-way streets, just like in a downtown area, so you can only go one way. But all the streets connect, and a lot of times they will move back and forth between different streets. The same thing with our metabolism. As part of homeostasis, we move these molecules back and forth all over the pathway and end up in different endpoints depending upon what our body needs. So in the American diet, let's say, yeah, we do need ATP, and we tend to eat lots of sugars, right? But does all of the sugars end up as CO2 and ATP and water? No, if you eat way, way too much sugar, what can metabolism push that all into? Where do all the carbons go? If you eat way too much sugar and you don't exercise enough, what do you make? Fat. They take the carbons, a potential energy of sugar, and they put it into a carbon base, into fat, because you've overwhelmed the system and you've made enough ATP, so there's no reason to break it down. I'm going to store it. And so all of these are interconnected and can go in different directions. Okay, there are a couple of one-way pathways that are important, like how I make glucose or sugar. And that is limited And so we may have lots of calories in our body, but our brain needs glucose, and we may not be able to convert things into glucose. And so we always have to constantly eat things that can be converted into glucose so that our brain functions. Okay? And so we're going to look at this. Now, each one of these pathways will be an entire chapter, okay, as we look at this. And we will go through the entire way. Now, ultimately, what we're really looking for is energy values. And we're going to look at reactions, and we're going to look at how much energy it evolves. And so we talk about delta G, and the prime naught up there talks about the standard free energy. And you normally look at kilojoules per mole, and you can see a lot of different reactions will give off lots of energies, okay? And so in this case, we're going to talk about taking off a phosphate. That's what this chart is, and it gives a lot of different values. Now, if we reverse the reaction, we have to reverse the sign of the energy, right? That's something in GChem. What the main thing is here is one reaction, and it's this one right here. Okay? That is a defining reaction. ATP broken down into ADP and PI has negative 30.5 kilojoules per mole. This is relatively a very high amount of energy. Okay? The breaking of an ATP will fund a lot of other reactions so that they can be made. And so that's kind of our make or break point. Most reactions for synthesis cost less than 30.5 kilojoules per mole. Okay? Because when I break an ATP, it'll allow it to go because of Hess's law. Two reactions combined, you add up all the energies for the overall process. Now, when you look at that, is that the highest energy, breaking of ATP? No, there isn't. There's some others that are bigger. All right? Those bigger ones are representative of places in which I can make an ATP. There are not a lot of them, but they're out there. And there are times in which I need to make ATP back, so some reactions give off more energy, and that energy can be used to create an ATP from ADP and PI, the diphosphate and an inorganic phosphate. And so we're all concerned about what will happen when the reaction occurs. We're going to be looking at the energy. This is the application of Hess's law that you learned way back in GChem, and we probably gave you really awful problems and gave you like 4 reactions, and you had to take a half of one and 3 1/2 of the other and flip some of them and change the sign, and we made you calculate. But here is where the application actually occurs in the body. We would like to place a phosphate on glucose to make it glucose 6-phosphate. Okay, but that is an energetically costly reaction. But we will then break ATP by hydrolysis, adding water, and that gives the energy. And when I add up those two processes, the overall combination of those two is negative delta G, which means it should be spontaneous. Although that's a misnomer, because they don't work independently. In the body, we're not going to have 2 separate reactions. They're actually going to react together in a specific sequence. Okay? Ultimately, it is the application of Hess's law, but we just do it in a little bit different order. And so a lot of reactions are coupled together so that the overall process can go. Now, the standard versus the actual. That prime naught meant that all concentrations are at 1 molar, the pH was 7, the temperature was 25 degrees Celsius, okay? And that's where all the reference material comes from. But really, the delta G is dependent upon what your real concentrations are. So there's an adjustment value that you have to make inside the body for real. And it's all dependent upon the equation. If you go back to GChem 2, which is, I know, traumatic for some people, going all the way back to GChem 2, the adjustment is RT, natural log of the concentration of the products over the concentrations of the reactants, and each of those products are raised to a power based upon their coefficient. Okay. What we can do is manipulate the reaction, and the standard delta G, it may actually be positive. Okay. But if we manipulate the ratio, okay, and we make the ratio below 1, that value, that RT natural log, can actually become a negative value. What we can do is we can drive the reaction, meaning that we can put a whole bunch of reactant and very little product, and we can force the reaction to go even though the delta G is positive, only up to a certain point. And so a lot of times the body will use that strategy too. It'll lower the reactants to kind of drive the reaction over. And so we have to take a look at what the real concentrations are to actually know what the real values will be. If I take a look at the energy for ATP itself, okay, there's a lot of energy, you know, negative 30.5 kilojoules per mole. Can anybody tell me where all of that energy comes from in the ATP molecule? Why does ATP like to be broken down? The energies are in the bonds. Where, in which particular bonds? Mm-hmm. Now, why is there a lot of energy? Why would it rather be broken apart than put together? Do you see something about the phosphates and the oxygens? What charge is ATP? It is a polar molecule, yes. Very polar. It actually is so polar it actually has charge. And so if you take a look at it, look at each of those oxygen in the phosphates. There's at least one oxygen, sometimes two, that have a negative charge. And they're really close together. Is that good? No, we think about opposites like to attract, right? Negatives or positives, if they're close to each other, want to repel. But they can't repel because there's a covalent bond holding them together. ATP is under a lot of strain. It's holding a lot of negative charge really close together. Breaking it apart is what releases all of the energy. Okay? That's why ATP has a lot of energy. Now, when you put it into the body, the one thing that we tell you once in Biochem 1 and never again, you're supposed to know it for the rest of your life, is that with every triphosphate, diphosphate, monophosphate in the body, there's one ion that's roaming around, and that's magnesium. Magnesium is a very important electrolyte. Do you see what magnesium is doing to the ATP up there? What is it doing? Yeah, it's really offsetting the strain. So what does the magnesium do to the potential energy of ATP? Does it make it higher or does it make it lower? Lower. Because what you're doing is, remember, the energy comes because the negatives are next to each other? Well, what if I put a positive in the middle? That stabilizes the structure. Okay? So the magnesium roaming around takes away a little bit of the energy. Okay? But it's always there. And it's very, very important. But remember, this negative log stuff, RT log of the concentrations, well, in every cell, the delta G prime is based upon 1 molar. Are these molar concentrations? Not at all. They're millimolar. They're much smaller, and they're not even. So when I put all of this in, I take away some of the energy because of the magnesium, and then I put some energy in because of the concentrations really inside the cell. What really ends up happening is the effective amount of energy for ATP is about -50 rather than -30. Okay, that's cool. I got more energy. Here's the problem. All of those other reactions that we looked in that table, they're also a little bit different. And here is what's good for us. If ATP went up 20, what actually happens is every other reaction in the body goes up about 20. Okay, not all of them, but just about every one. So that allows us to do this. Looking at the standard energies, almost always, if we just use standard energies, it comes out to about right. Okay, because we're taking differences. Every so often, the concentration does do something differently than the actual standards, but not so much as to, I mean, it's like this. If you get it a 98% right all the time, if you could walk into a test and make 98s every time you sit down, would you be okay? Okay. Almost everybody in class will say, okay. There is that one person that says 98's not good enough, right? And so we tend to use the standards. But I want you to understand that the reality is just a little bit different. And every so often, someone doing real research might have something come out not what they expect, and then they would have to go down and investigate this to see if the concentrations were what was making it deviate from what they expected. Okay? Now, a lot of times we talk about phosphate bonds, and they use, instead of actually drawing out the phosphate, they use a little circle with P. And what it really is, there are some high energy compounds and low energy compounds. Essentially what they're doing this, they're making ATP as the standard, and anything really kind of above that is a high energy bond, very limited number. And most everything else is a low energy bond in which ATP could fund the reaction. And so one book puts it this way, that this is kind of the standard point. And so that's where I want you, kind of like pH is always, we measure at 7, and that's kind of the reference point. All biological reactions inside metabolism have like kind of a set point as ATP, above it or below it. And that's kind of we kind of look at it that way. Let's look at some of those high energy reactions, okay? There are 3 described, one that has 49 kilojoules per mole, one that has 48, and one that has 61, okay? I want you to look at these 3 reactions, talk amongst yourselves, your neighbor, and tell me the principle that you see in these reactions that might account for a lot of energy. What do you see represented in those reactions that may account for energy? It's a concept that you should have recovered in organic chemistry, but I don't know how pointed he might have pointed that out. Now, each one of these, I'll tell you, each one of these is losing a phosphate, so that is not the problem. That's not where the real big energy is, although losing a phosphate gives energy. Okay, there's something else in these reactions that give us more stability of the reactants than more stability of the products than the reactants. Okay, so other than feeling lost, what do you see in this? What do you see in this first reaction here? Okay. Reactant, right? Phosphate, right? Phosphate falls off. Okay. That's common amongst everyone. Then you get this compound, right? Then they draw another compound. What does the next compound show you? Resonance structure. Okay. This compound, that phosphate falls off. I get this compound, right? Why do they draw another compound? It is a what? Resonance structure. This one is hydrolyzed, and this one is a special resonance structure called a tautomer, okay, which means that it's not just it's an equilibrium, but it's way over here. So common between the 3 is the ability to form a resonance structure, the final product. Now, one of the reasons why phosphate, when it falls off, gives off a lot of energy is it creates resonance structures. That oxygen, or that negative charge, now can resonate through all of the oxygens, and the more resonance structures you can draw, the better. meaning that more energy stabilization. And so all of them have the phosphate falling off, making resonance structures, but the other part of the molecule also can draw a resonance structure. So you get two entities that can draw multiple resonance structures. The resonance is where the energy comes from in a lot of organic reactions. That's where you get a lot of stabilization. Resonance is very, very important in terms of making a stable intermediate. We tend to really like that. And so with the really high energy ones, not only does the phosphate make a resonance structure, but also the other component makes a resonance structure. Okay? If it's a lower energy reaction, you can't make another resonance structure. Okay? All right. ATP is going to be used for a lot of things. Okay? There are 3 phosphates. They are labeled alpha, beta, and gamma phosphate, depending upon their relationship. The closest to the ring, the ribose ring of ATP, is the alpha, and the furthest one is the gamma. Now, you can do a nucleophilic attack at each one. You can attack the alpha, you can attack the beta, and you can attack the gamma phosphate. All right? Now, the reactions and the mechanisms are going to be important for you to think about, but you probably never have thought about that too much in the past. And a lot of times, even when they write it in the reaction that you see in the book, they don't tell you where it attacks directly. They tell you absolutely indirectly. Okay? And so what would happen is, if you attacked at the gamma, the reaction would give these products. ADP and 1 phosphate would move. Okay? So at the end, when you see reactants and products, if the product contains an ADP and 1 new phosphate somewhere, I know exactly where the reaction attacked. It was at the gamma. On the other hand, the second most often one that is reacted is the alpha. And if it attacks there, what falls off is 2 organic phosphates. So what falls off is that portion. And what gets added is 1 phosphate, which is that phosphate, the ribose sugar, and the adenine base. Okay? That's the second most often thing that is attacked. The third is an attack at the beta. This is really rare. The beta will have 2 phosphates attached to the nucleophile coming in, and AMP is the leaving group. So if you just look at the products, you know, did I attack at the alpha, at the beta, or gamma? I have ADP if it's gamma, 2 PIs if it is alpha, and an AMP if it's beta. Okay? So they give you all the information, they just give it to you hidden. Okay? And so this will be important as we look at reactions. Most often, you see this is 1. You'll see that every so often. Very rarely, and I don't know why, but very rarely will you ever see the attack at the beta. It can occur, it just doesn't, it's not optimized for a lot of reactions. All right. What I want you to understand is how they write reactions. And so if I take a look at this, I have a reactant, glutamate, and I have a product, glutamine, and I have a amine coming in, which is ammonia, and this. And then they say ATP is part of this reaction, and ADP and PI is the end product. All right. How many reactants are in this reaction? 3. Very rarely, if ever, do 3 molecules react at the same time. It's usually 2 react, I form an intermediate, and I react the third. Okay? But a lot of times, for brevity, okay, inside a textbook to make it shorter and fit in a page, they will write this and they will expect you to understand what's going on. ATP is an energy molecule, okay? And so a lot of times this molecule is going to be termed, it's going to activate the reactant, okay? Meaning it's going to make somewhere on that molecule a wonderful leaving group, because we need a good leaving group to facilitate the reaction, okay? And so what happens normally is one of the two species reacts with the ATP first. And if I take a look at the product and do some inventory, I see the phosphate is on this oxygen here. All right. Also, because I know it's ADP and PI, I'm going to attack at the gamma. So this oxygen is going to react with the ATP first to create this intermediate. This right here, a phosphate, is the most wonderful leaving group that you can have. One of the best. Okay. The next thing that happens is the nitrogen will react with this carbonyl. Electrons kick up, kick down, and phosphate will be the leaving group. And I'll end up with a glutamine where I have the amine attached to this carbon where it once was attached to an oxygen. Okay? And so a lot of times there will be an intermediate that is, this right here, this intermediate here is the activated one. Meaning that if I attack that, it's going to have a wonderful leaving group. And so that makes it more likely to be reacted, and it makes it more likely to go to full completion. What is really strange about biological reactions inside the body is the yield. How many people have taken organic chemistry? Lab. Okay, you've taken lab. What was your normal yield for your reactions? Would you want your yield as the grade? No, what were you getting yields? What percentage? 50? That's if it was wet when you measured the product, right? Organic reactions in an organic flask really don't go to completion a lot of times. They have really low yields. The reactions inside the body are 100%. Okay? We do it a little bit differently. We will do the same organic reactions that you can do in an organic flask, but we do it in a totally different pathway. We do it actually inside an enzyme, which forces the reaction to completion. And what we are going to do is make wonderful leaving groups. We're going to optimize the reaction so that it flows to 100% completion. And so totally a different type. All organic chemistry going on, but in ways that are controlled, whereas in the organic flask, for you to get a reaction, you heat it up. You make them bounce and hit each other with more force, and they can hit each other in all sorts of different random ways, and you get side reactions. Now, when you take a look at your percent yield, that doesn't mean all of your reactants haven't reacted. Most of the time, every one of your reactant has reacted, but you only made 50% of what you wanted, and you made 50% of byproducts that are not what you want. What we control in the reaction inside the body, inside the enzyme, is we make almost no byproduct. That is not the one we want. And so we can control that reaction. Okay. ATP, I told you that the example that we just used was an attack at a gamma phosphate. Here's an attack at the alpha phosphate. Okay. And so when I attack the alpha phosphate, the two PIs get broken down. That actually releases energy. Plus the AMP, which is attached, can fall off, and that is a good leaving group. And so if I break the ATP at the alpha, I get more energy than when I break, because I break two phosphate bonds as a result. It's kind of like more bang for the buck. I use one ATP, but I get a lot more energy than the 30 value. All right? Even in the process of adding, in this case, something to that long fat, so I had to use up energy, it even had more energy than what ATP would be by itself. And so that is great. It's a way to use ATP, but I end up with AMP, which is a very non-energetic molecule that has to be made into ADP and then back up into ATP before it gets used. So we do elicit a lot of energy, but then it takes a lot more energy to get it back up to ATP. But this is seen quite often to drive reactions that are very, very energetically unfavorable. That's why we use that alpha attack. All right, before we get to our first pathway that basically everybody starts with, which is glycolysis, we need to take a look a little bit about sugars, okay? And so sugars are, or you can call them carbohydrates, are a molecule that has a ketone in it, either an aldehyde or a ketone itself. So it has a carbonyl, and it has hydroxides attached to it. And so in this case, the first one up at the top is glucose. The second one up at the top is fructose. One is a aldose, or contains an aldehyde group, and one is a ketose that contains a ketone group. And those are the two versions that we can have, aldose and ketose. Every numbered, okay, one has a different name, and unfortunately for you, every different stereoisomer of those oxygens, hydroxides, whether they are in one position or the other, changes the name of sugars. And so there are a ton of sugars out there. Glucose is the one we talk about because glucose is the one that is the majority one that is used within our metabolism. Okay? And so when we talk about carbohydrates, there are a ton of carbohydrates. But when we eat them, most of them get converted either directly into glucose or into one of the derivatives of glucose. And so we kind of think we can eat lots of sugars, but they all kind of funnel down into those ones that come that are part of glucose pathway. Glucose also is found both in the straight chain form and in the cyclic form. In actuality, if you go to the stockroom and get glucose and you put it into water, you will have an equilibrium between the cyclic and the acyclic form, open chain and closed chain, and it will equilibrate over time, and it'll get to the best thermodynamic structure. We write this sugar a lot of times like this, but way back to organic chemistry, you know that sugars can take two forms, the chair and the boat, or manipulations of that. Of this, the chair and the boat, which is more stable? Chair. So in the solution, it'll equilibrate to get into the chair conformation, because when glucose is in the chair conformation, all the hydroxides are in the axial positions, and all the hydrogens are in the equatorial positions, and the equatorials are close to each other. So the smaller ones that are you want the smaller ones and the ones that are closest, so they have less interaction. You want the big ones on the axial to stick out so that they don't interact with anything. And so glucose in the cyclic form, even though we write it a lot of times like this, you have to think about it, it is actually in the chair form almost all the time. And if it's not in that chair form, it can be in the open cyclic form. And so we need to understand that. And then that six-membered sugar, when on the ketose form, when it makes the cyclic form, it has a five-membered ring. And so we either have a pyranose ring or a furanose ring. All right? Sugars. Unfortunately, they have stereochemistry. Okay? And so a lot of times in organic chemistry, we talk about RNS. Nah, that's not what we talk with sugars. We call them D and L. Okay? And the D and the L designation is what we use, but we also talk about D and L, little d and little l in organic chemistry. Do you know where the little d and the little l are referred to? What instrument do you use to measure little d or little l? It's a rotation. You put it in a Logan tube. You hit it with polarized light. You look at the end and you have to twist it. I don't know if you actually did that in organic chemistry lab. It was a polarimeter, And the little d and little l refer to, did it go left or did it go right? And those little d and little l has nothing to do with big D and big L. The big D and big L are reference to glyceraldehyde, our most simple sugar. It has a ketone and 2 hydroxides. And in the Fischer projection, if it's written in this form with the hydroxide on this side, it's called D. And if it's on this side, it's called L. And the ketone is always at the top by definition. And so that's where they reference it. The D and the L will be the last hydroxide furthest away from the ketone. Okay? So there could be lots of hydroxides. The DL conformation is the furthest away. All right. Now, simple sugars are great, but you don't eat simple sugars. Okay? You don't actually eat glucose by itself. That's kind of a misnomer. You actually, the smallest sugars that normally you eat are what are called disaccharides, 2 put together. But our body processes monosaccharides, single ones. And so the simple sugars can react together, and they react at that ketone. Okay? And so at that ketone, we can have an alcohol, which could be another sugar itself or any alcohol, reacts at that ketone to create, if it's an aldehyde, what's called a hemiacetal, or if it's a ketone, it's a hemiketal. Okay? And there's just a reaction that occurs. Many of the sugars are linked this direction or this way, where you do that. Okay? When we take a look at that sugar, I want to point out one more thing. All right? Here's D-glucose. It can form the cyclic form, and I have 2 versions. Okay? I have 2 versions. One is called the alpha, and one is called the beta. Okay? If I have 2-handed molecules, it's called an enantiomer, right? You have multiple, you can have diastereomers. Okay? Here, we take a look, and the difference between these 2 are called anomers. What's the only difference? The position of that hydroxide. And this hydroxide, either right here or right there, was that ketone oxygen. Okay? It can either be above the ring or below the ring. Above the ring is called beta. Below the ring is called alpha. Okay? The beta for glucose is more stable because the beta carbon puts the hydroxide in the axial position, not the equatorial. The alpha puts it in the equatorial. And so the most stable conformation and distribution, if you put it just in water, is beta D glucose, okay, or beta D glucopyranose ring. And that's the most stable version. All right, but important, we will use the term alpha and beta later with other things. Another way that people have written the sugar is in what's called a Fischer projection. And check out the bond for for oxygen. That is a big bond. Okay? Why in the world did we ever draw these? Okay? Now with computers, I can draw that real simply. Okay? Way back when you used a typewriter, no way could you draw that. But with a typewriter, you could have made lines like this, and so you will see this written. The only reason why they did it way back when was because in a typewriter, I could actually display a ring this way. And so you may find it in older books. People still use it. But we see this, or we see the true chair boat form. So I don't want you to be confused if you see that. Okay. Here's the boat form. Here is the example. I told you beta D is the most stable. Axial, 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 axial. All the hydrogens are equatorial. That's the reason why. Okay. I have no idea why I told you twice. We called one ring a Paranos ring that had 6 members and a Furianos ring for 5 members. We actually do that because of these organic structures. Okay, pyran and furan, they are 5 and 6 membered rings with 1 oxygen, and so that's where the name comes from. You might actually have seen this one right here, furan. It's a solvent that is used quite often in organic labs. Pyran is not used as often, but this is where that name came from. And so they just didn't totally make it up, although they made a derivative from something else. All right. The glycidic bond, where we react at the anomeric carbon, that forms hemiketals and hemiacetals, can be converted into full acetals and full ketals to actually make linkages. The reason why we introduce these is because we look at disaccharides and polymers. All right. Here are some really important ones. Lactose. Okay, what you need to know here. Lactose is a galactose sugar and a glucose sugar. The connection between them is a beta. All right? It is a beta that becomes, meaning this, okay, they have it all in one line here. If this bond was true, what you would have is an oxygen there and the sugar above it. So the second sugar, the glucose, is above the ring. That's why it is beta. And it's between, this one is the 1 carbon, when we start numbering all around. This is the 1, 2, 3, 4th carbon. So it's a beta between 1 and 4 linkage. Okay? So if you see that little cross hatch, that's a weird bond. That indicates beta in this book. Here, in sucrose, the oxygen's kind of down in a V. So the bond is going below the ring. This is called an alpha. And again, this is a 1 as I go the ring. And on this one, okay, that's the 1 carbon, that's the 2 carbon, the way that it's put together. And the last one is 2 glucose put together. It is alpha, and that's the 1 carbon, and that's the 1 carbon, how they're put together. All right? Here's the problem with sugars. I can put 1 and 4 together. I can put 1 and 3 together. I can put 1 and 5 together. I can put any of them together. I can put 2 and something together. And these all become important later, but it gets so complex. It's not just 2 that can become, but it's 2. Is it 1, 2 alpha? Is it 1, 2 beta? Totally different. If it's 2, 1 alpha, 2, 1 beta, totally different. Has a different 3-dimensional shape. It is used by the body differently. And so sugars is a very hard thing to study because the diversity is so large. And we look at 3-dimensional shape. It's not that there's the 2 sugars, it's how they're oriented and where the hydroxides are, create a fully different 3-dimensional structure, which is recognized by the body totally differently. And so the disaccharides are important because we eat a lot of these. Okay? Sucrose. What is that? Do you know a common name of what sucrose is? Oh, you do, but you don't. Nobody knows? No, it's not Splenda, but that's a good guess. This is one of the, Splenda is a derivative. This is natural. This is sugarcane. This is the natural sugar, that you would buy in a big bag. Okay, this is what plants make as one of their products of photosynthesis. A simple, what we call simple sugar. A lot of times when we're thinking biochemistry, though, we think, oh, all sugar is glucose, and we think that bag is glucose. It's not. Okay? If you're actually in the medical field, sometimes in an IV, we do give glucose directly to patients, but they don't call it glucose. They call it dextrose, and a dextrose bag. I don't know why they call it dextrose, but that's the only time that you would ingest glucose directly, and they're giving it to you IV because your body's under strain, and they want you to have a quick dose of glucose in your bloodstream that doesn't have to go through all the biochemistry so that you can feed vital organs. Okay? But very rarely do you ever eat any single sugar. This is probably the most common that we, especially in the American diet, that's the most common. Where do I find lactose? You've heard that. You've heard lactose too, right? Dairy. Okay. You've also heard of a disease with lactose. What disease? Do you know? That's right, lactose intolerant. That is a disease state. The enzyme to break lactose is lacking inside of you. Okay. Unfortunately, in the American biochemistry, we tend to have lactase, the enzyme that breaks down, ACE enzymes break down things, so lactose is broken down by lactase. We have it when we're young. But a lot of people will lose the ability to produce that enzyme, and that creates problems because disaccharides cannot be absorbed. They should be broken down in the intestine, right? And then single sugars can be absorbed through the intestinal wall into our bloodstream. Normal process. But what if you eat something that has lactose and you can't break down lactose? It can't be transported. Then you get a problem with osmotic pressure. Okay? What happens is, on either side of a semipermeable membrane, if something is in high concentration and something is in low concentration, what the body wants to do is have the concentrations exactly the same. So what happens is if you have lactose highly in the intestines, water rushes into your intestine. That doesn't feel good. And then you get watery intestines, makes things flow a little bit too much. And so you get cramping, you get diarrhea because you couldn't have broken them down. The body's trying to dilute it, and that causes a disease state. It's the absence of an enzyme. What's a solution to someone that is lactose intolerant but wants to drink milk? Not just skim milk, because skim milk only gets rid of the fat content, not the sugar content. But it is a certain kind of, well, you can go with an alternative that has no lactose, yes. But what if you wanted cow's milk? There is a product called Lactaid. And what they do is they take a lactase enzyme and they just treat the milk. And they break the two sugars down into glucose and galactose already in the carton before you drink it. And someone who's missing that enzyme can still absorb the single ones. And so you can either treat it or you can take a pill with the ingestion, and that pill has the enzyme in it, and so you supplement the enzyme as you're eating what you're doing. And so you can have Lactaid drug, or you can buy that. And so that can get around that particular disease. But these are usually eaten, okay, in the stomach under the very intense acidic conditions. It doesn't actually break it down. It needs the enzyme that is in the intestine when they're broken down and then absorbed at that moment in time. And so, I have on this, the sucrase is broken down, the sucrose is broken down by sucrase. An enzyme that has an ACE is something that breaks things down. Lactase and maltase are the ones that break them down. Oh, lactose intolerance. Okay, we already talked about that. All right. Now, in this, there's a lot of different storage molecules, and what we're going to find is one of the most often seen polymer of sugar is lots of glucoses put together. Okay, long strains of glucoses. They call those starches. Okay, you may think about starches when you eat. Okay, And starches, when you eat those, tend to be easily broken down, create lots of sugar. This then increases your carbohydrate intake, and then if you don't use all that energy, it becomes fat. But oh, does it taste so good? All right, you've got lots of breads, potatoes, even a good vegetable like corn. You know, all these starches are just long chains of glucoses. The glycidic bonds are called oglycidic because there's an oxygen between the two sugars. All right, and in this case, it is an alpha 1,4 in this. And so one kind of starch that is in plants are called amyloses, and they're broken down by an enzyme called amylase. That sounds pretty straightforward. And it breaks them down into maltose, the disaccharides, and then we know the disaccharides can be broken down into monosaccharides. And this can actually, amylase, the first time you see this enzyme is in your saliva. And there's a very simple experiment that some elementary school kids do. If you take a saltine cracker, okay, and you chew it up for more than normal, okay, most people take a bite and then chew it up and swallow it. But if you actually continue to chew on just a simple cracker, saltine, it'll actually start to taste sweet because the work of the amylase will start breaking down into sugar molecules that will actually interact with the sweet receptors. The whole starch doesn't. It's too big to fit into the binding spot. But when you break them down into disaccharides and monosaccharides, they can actually bind, and you can actually taste the sweet after the enzyme starts working on it for a little bit. And so just you have to chew it for a long time and allow the enzyme, the process to work. And so this is one of the first enzymes as part of digestion. It works on the starches that you eat. And then there's digestion that happens in the stomach. There's separate digestion that happens in the intestine. Okay. All right. Glucose is what we're really trying to get. Okay. And we're going to degrade this. Now, I told you ATP concentration, when we broke it down, 30 kilojoules per mole, right? I said that was big. If you break down glucose, how much energy do you get? Bigger than 30, right? A lot bigger than 30. That is a huge potential energy, okay? 2,840 kilojoules for every mole, okay? You could actually calculate that into calories, okay? But if you think about, let's say, sucrose, sugar, okay? 1 mole of sugar is something like 500 grams or half a kilogram. And every kilogram is like 2.2 pounds. So that much energy is present in about a pound of sugar. Okay? That's how much energy you could potentially use. All right? If you eat a pound of sugar and you convert that over, that's how much energy you would need to exercise to burn it all up and not store any fat. Okay? So, yes, it's a wonderful big amount, right? But in terms of our daily caloric intake, for you to maintain weight, you got to use at least as much energy as you are burning. If you intake too much, you store. If you intake too little, you could lose. Okay? And so there's a potential amount of a lot of energy when we take a look at this. There's something about this particular number here, okay? It's under what heading? Aerobic. What does that mean?
Uses oxygen.
Yeah, it uses oxygen. All right. So remember, combustion is combustion of a hydrocarbon is oxygen plus this. If we can use glucose, and ultimately the reaction is with oxygen, although through multiple steps, then I can get that much energy out. It's very, very energetic, okay? But we can actually get into a degradation event called the anaerobic degradation, okay? And in general, an anaerobic is called fermentation, okay? And we use fermentation, especially of glucose, in two ways. One inside the body, and then one for a very profitable reaction in which yeast will convert sugar in an anaerobic form, and the outcome is ethanol, okay? Inside our bodies, the fermentation process, we just don't have that enzyme to convert it into ethanol. I think that would be really, really bad, because right now when you do anaerobic workouts, what happens in your muscle? What happens if you do anaerobic workout? Do you know? You're not processing and you're not extracting the energy the way that you do, and you create a byproduct, lactic acid. Okay, lactate is the ultimate, all right? And so lactic acid burns. It tells your body you're not using energy efficiently, and it kind of stops you. Just think if we had the other enzyme pair. What would happen when you worked out really, really hard? You would get drunk. Maybe more exercise might happen, but that might not be the best for us. No working out and driving, you know. And so, kind of a different process in terms of what we are looking at. Now, glycolysis, you've seen this in multiple ways. You've probably seen it in high school, and you've seen it in biology, and you've seen it all over the place, okay? And a lot of times I see it in biological handbooks or pictures. You've got 6 balls strung together, and they will break into 3 triangle, you know. We're actually going to look at the structures because this is biochemistry, and so we're going to look at what the enzymes do. It is a series of 10 reactions, okay, and we will look at 10 enzymes that will process this, and we'll see the intermediate molecules that we'll be converting into, all right? And so here's the obvious question. Yes, you need to know the structure and the name of each one, okay, as we go through this. The reason is, remember that in metabolism it's this big old connection of roads, right? Well, many of these molecules will be connectors to other things. And so we're not just learning glycolysis for itself, but we're also going to see where the crossroads happen, okay? And so in this, traditionally it's broken into 2 pieces. The investment part, where I use ATP, or called the prep part, and then the payoff part, where I'm going to get ATP. It's like this. To get money, you have to spend money. To get ATP, you have to use ATP, okay? And so we're going to eventually oxidize this all up, and you saw that numeric value. But what that will translate into, if all of the energy, potential energy in that glucose is converted into ATP, the most that you can make in modern textbooks, you can find other values other places, but is 32, okay? That's the maximum number of ATPs you can make at the end, although 30 is probably the most often one seen. We'll talk about that later as we go through several chapters. All right. In this process, we were going to use 2 ATPs at the beginning, so net minus 2, and at the end we will make 4. And so this glycolysis makes 4 of the possible 32 ATPs. It isn't really a lot of the end product that we want, but we make other piles of energy in different forms that we can account for, all right? After the 4th step, okay, after the 4th and 5th step, we actually create two of the same molecule. So starting in the sixth reaction, you're doing it twice, okay, as you go through there. Also, look at the reaction. There are some that are single arrows and some that are equilibrium arrows, all right? Single arrows tend to be the ones that use ATP. They're so energetically favorable in one direction that there's really no equilibrium back. Others can go back and forth, just depending upon what the concentration is of, do you have a lot of product? Then it'll go back to reactant. If you have a lot of reactant, it'll go to product. And so there are some key differences in each one of those reactions. So we're going to look at these in very much detail. Okay. Now, once we get to pyruvate, which is the end. Oh, sorry, yes.
If you said that
Huh?
you use 2 ATP in the beginning and then you make 4, is that a net of 2?
A net of 2. We only make 2, yeah, as a net, which is a very, very tiny amount of the energy. Now, the last molecule are 2 pyruvates. That's the end of glycolysis. That is still energy, because any carbon that has hydrogen attached to it or hydroxides attached to it can still be oxidized. If we want, under aerobic conditions, we can get to 4 CO2s and 4 waters, and we produce all of the ATPs we can get from that particular glucose. We can convert it in yeasts to ethanol, okay, one of the most important biological reactions that have been used for a very, very long time. If we exercise too hard and we run out of breath, meaning we can't efficiently get oxygen to our muscles, we'll create lactate. And then there are some other fates. Pyruvate can be used to make some amino acids when we synthesize, and we'll introduce some of those. So pyruvate is kind of this break point where a lot of things can go in different directions. So pyruvate is a key structure where a lot of things can happen. Okay, and so we're going to also create energy in terms of ATP, and we're going to create energy in an electron acceptor. Okay? Really, we're doing oxidations, and oxidations is transfer of electrons. Redox, I give and take, lose electrons. And so we're going to have energy in the form of NADH is an energy molecule, and it's an electron carrier, and we're going to convert some of the energy there. Okay? The overall energy process of glycolysis, this is the overall reaction. Glucose plus 2 NAD plus 2 ADPs plus 2 PIs equal 2 pyruvates, 2 NADHs plus 2 protons plus 2 ATPs plus 2 waters. Okay? And if I take a look at all of the energies and I add them all up, I give off minus 85 kilojoules of the minus 2,840 or 60. It's huge. That is a very tiny amount. But what we're doing is we're converting molecules into structures in which we can extract more. This is just one stage of a basic 3-stage process to get to ATP. One thing that we need to look about in terms of glycolysis is that all of the intermediates are phosphorylated. Okay? That means a phosphate's added to it. The phosphate at normal pHs within the cell means that all the phosphates are negatively charged. The negative charge traps molecules inside the cell. Okay? They also, once things are phosphated, carbons are phosphated, a lot of times that kind of links them to or tags them for the metabolic process. Okay? These small molecules. Phosphates also carry around magnesium, and so many of the enzymes is dependent on the concentration of magnesium in the system. And so making sure that you have enough magnesium in your system is important to make sure that your metabolism is actually functioning correctly. All right, here is the first step of the reaction. Glucose will create glucose 6-phosphate, or G6P. Okay? Here's the bad thing about biochemistry. We're lazy, so we use acronyms, and it's going to get crazy, the acronyms, all right? And so I just warn you now, that's what's going to happen, all right? So this is the first reaction. You can see that one hydroxide, and when you look at reactions, you kind of do inventory. So if I look at the inventory, right around here, both reactant and product, nothing happened. Nothing happened. Nothing happened. This oxygen here is that oxygen there, and then something happened. Now in the book, it put a box around it. That's really nice. Won't put a box around it on the test. Okay. All right. So you can see that the reaction actually is right here. Okay. And so glucose and the other reactant is ATP. It is done inside the enzyme called hexokinase. Okay. Kinase, an enzyme that adds a phosphate using ATP. It's a generalized enzyme class, and the hexo comes because it has 6 carbons. Okay, and so there's an alternative enzyme called glucokinase. It's going to kinase, put a phosphate on glucose. It's found in a different tissue type. The majority of sugars are phosphorylated by hexokinase muscle. That's where we're going to find that. Glucokinase is found in the liver. All right. All right. So what I did was I drew out ATP up here. Okay. And so instead of just seeing ATP, there we go. Now, looking at this, here's the reactant. One phosphate is transferred, and ADP is the product. Which phosphate do we attack? Alpha, beta, or gamma? Gamma. All right. So I'm going to articulate a little bit more here. All right. I'm going to have a proton. Okay. I'm going to articulate that proton there on the hydroxide. And there's a bond of electrons between the hydrogen and the oxygen. All right. Here's a key for enzymes. And you may have learned this in cell. I did teach it in Biochem 1. But inside the enzyme, all the reactants bind inside the active site. Okay? It's a small little hole or cleft that creates kind of like a very specialized beaker where the reaction is going to occur. What happens inside the active site are 2 things. It aligns the reactants perfectly together for the 2 things that are going to be reacted right next to each other. Okay? And so it gets them close. The second thing it does, it will stabilize a transition state, the high energy state that is the energy barrier for the transition. Okay? The third thing that it does, it provides protons and bases to allow your reaction to occur. Now, in an organic flask, inside a round bottom flask, you learned organic mechanisms by an acid catalyzed pathway or a base catalyzed pathway. Because when you drip in a reactant, you can drip in acids or you can drip in bases. If you drip in acid and base, what happens? What does the acid and the base do? They neutralize each other, and so you don't get it. You don't get any catalytic because now you have water and salt, and that doesn't help. But inside an enzyme, you can have in the same reaction a base being worked on and an acid being worked on, okay, available. All right, I'm going to show this way. Now, one of those resources that I want you to look at explains this in a little bit more detail. But I'm going to show bases like this. I'm going to show a base B with a lone pair. Okay? Essentially, what this will be is one of the R groups of the protein, one of those R groups that we learned, it will act like a base. Okay? And then I can have a proton anywhere, and that will be an amino acid. And I can write a proton as H+. Okay? And so, in this mechanism, the first thing I look at is, in the reactant, I have an extra hydrogen that I don't have a hydrogen in the product, right? The hydrogen to the oxygen. So I will write the mechanism that says this base takes that proton. Okay? If I take that proton, then the electron pair between the hydrogen and the oxygen is now free to roam about the cabin. All right? Now, in organic chemistry, what you would write is the oxygen is negatively charged. That bond would sit on the oxygen. A negative charge on the oxygen is high energy, but it's not so bad. Why? What is a characteristic of oxygen? It has high what? Oh, you're going to have to go all the way back to GChem 1. There's a trend as I go up. Electronegative. So it likes electrons. An oxygen with a negative charge isn't bad even though it's high energy. Okay? The enzyme stabilizes that, but also the enzyme doesn't allow that negative charge to be on that oxygen for a while. Inside the organic flask, if I deprotonate it, that negative charge sits on that oxygen until it floats around and finds what it needs to react with. So we actually formally write it that way and show it as an intermediate. But because we're in an enzyme, that negative charge is going to be right next to where it's going to react. So what we do is we show that as this gets taken, this particular pair of electrons can attack because it's right there. Okay? Anytime that we attack a carbonyl, okay, the pi bond must kick up on the oxygen as a transition state. We also do that at a phosphonyl. And so the phosphonyl kicks up so that the negative charge is sitting on that oxygen. That is the high energy transition state. I put an extra negative on that long ATP that has lots of negatives. The enzyme stabilizes that particular transition. It is high energy state. It's the excited state. Does things stay in the excited state? No. It would be better if it falls down. And so it kicks up and then it reforms the double bond. Too many bonds to phosphate. I can have 5, but right now I have drawn that it has 6. And so one of the bonds has to leave. ADP will be the leaving group, and I do it this way. This pair of electrons will fall onto that oxygen, and what leaves is the adenine, the ribose, and 2 phosphates, or ADP, falls off. This phosphate is added. This oxygen was the original oxygen of the hydroxide, and that phosphate was originally part of the ATP. And we could, if we label the oxygens, we could actually see that being the case, that where the oxygen is. Okay? I've created glucose 6-phosphate, the first reaction. All right? So I'm going to show you lots of enzymes, lots of mechanisms. Okay? Here's the deal. We're going to look at so many enzyme and mechanisms that you can't memorize every one of them. But what you can do is recognize we're going to see a lot of repeats. The body is optimized to do reactions at a pH of 7, at 37 degrees body temperature, in aqueous conditions. Okay? If you put this organic reaction inside a reflux at 37 degrees, at a pH of 7, in water, it would never react, because the energy barrier is too high. But inside an enzyme, that transition state is stabilized so that this reaction can go. Okay? And so, here's our first step. So there's only a certain number of ways enzymes get around this. So they tend to repeat. So we'll see some repeating things. You can't memorize everything. But what did I do in this first reaction? I took inventory. I looked at the reactant. I looked at the product. What is different? That's the only place you have to focus on, is where the reaction is occurring. Don't let the size of the molecule scare you, because most of it doesn't really care. In an organic chemistry textbook, what they do is they just put R, and you forget about it, and you focus in on what you're looking at. Well, here I give you what the R is. You just have to forget about that part and focus on what's going on. Okay. The second reaction, okay, is an isomerization from an aldose to a ketose. It's done by phosphoglucose isomerase. Isomerase changes the structure, okay, but if you look at the beginning and the end, the reactant and the product, there is no change in the number of atoms, the same number of protons, the same number of oxygens, the same number of carbons, same number of everything, just rearranged. Okay? Normally, the mechanisms for these isomerases is a series of deprotonate, reprotonate, deprotonate, reprotonate, deprotonate, reprotonate, until you get even, and it will end up being, you can convert one structure to the next. Okay? I also go from a 6-membered ring to a 5-membered ring. All right? And that is kind of weird. Okay? And so what we're going to find is we're going to have to do something a little bit different. Okay? And so the reaction starts by this. I have to open up the ring. Okay? So G6P, glucose 6-phosphate, and F6P, fructose 6-phosphate, are both sugars. That's the beginning and that's the end. I'm just going to show 2 intermediates. Okay? So this is kind of a little bit longer, okay, to get through those 2 intermediates. And so the first thing that happens, is what? What actually has to happen? Okay, so if you do inventory, right here to right there, okay, what is different between here and there?
Oxygen in the ring grabs the oxygen.
So this oxygen in the ring has to end up having a hydrogen, right? So that's different. Anything else? That bond is not there anymore, right? That's different. What else? Yeah, there's a double bond right here between the carbon and the oxygen. That's right there. And on this one, what is it missing? A hydrogen. 80% of the time when we do mechanisms, the first step in a mechanism is to deprotonate. So most of the time we're looking for which hydrogen goes off. And so I always have an acid and I always have a base. So I'm going to put an acid and base, no particular place. Base right there. I'm going to put an acid right there. Now what the enzyme does is it puts the acid and the base right in those positions where the reaction should be. Okay? So what do you think the base does first?
Attacks the oxygen.
Does it attack the oxygen directly? It's going to attack the hydrogen on that oxygen. So take that off. When I take that off, I have a lone pair of electrons. Where does it go? It forms the what? The double bond. But that's a booger, because when I form that double bond, I have now 5 bonds to carbon. If you did that on Chem 21 or another homework site, you get a big red box, right? So something has to leave. Which bond leaves? The bond right here. That leaves and picks up that proton. Okay. Articulate this. That last bond leaving. I could have said that it went onto that oxygen for a temporary time. That oxygen was negative, and that picked that up. Okay? But because it's inside the enzyme, the proton is right there, so it actually happens as a series of events, and they're all together in one kind of movement, one after the other. All right? So 1, 2, 3, and I've formed my intermediate structure. That's not where I want to be, okay? And so when I compare this intermediate structure and this intermediate structure, okay, what has to happen? Okay. Isomerases are always deprotonate, reprotonate. Do you see that first step? I took off a proton, and what did I do? Put one on. This is going to be the same thing. I got to take off a proton, and I've got to put one on. You can see right here, this oxygen has no proton, right? Here it does. There's a double bond there. There's a single bond there. There's a hydroxide. That's a hydroxide, no change. But what happened to that carbon versus here? It's lost one, right? Lost one, gain one. Okay? What do you always have to use? A what and a what? A base and a proton. And so I will take this proton. This pair of electrons will form that double bond. That means there's too many bonds of carbon at this position right here. So the leaving bond is the pi bond. That picks up that proton. And I get to the second intermediate. Okay. Now it gets a little weird. Okay. I have to find one that is deprotonated, and I got to find something that is protonated. Okay. What gets deprotonated? The oxygen that is located right here. That's the one that's losing the proton. Where's the entity that gains a proton? Carbon. Yes. And the carbon would be that one right there. Because right here it has no hydrogens, right? And here it has no hydrogens, right? It has a hidden one. Okay? Because I've got 3 bonds, but in a structure with 3 bonds to a carbon, the 4th has to be an understood hydrogen. So that was kind of hidden. Okay? So I've got to deprotonate this. I've got to protonate that one. Oh, no, that's not true. No, I don't protonate this one. I protonate that one. I'm sorry. Watch this. Let me see. If that is numbered 1 and that is numbered 2, this one is 1 and that one is 2. Do you see where 1 has 1 proton at the beginning and 2 protons at the end? Okay, so that's what we're going to do. All right, I always have a base because it's all about the base. Okay, so I'll take this one off. That pair of electrons, this oxygen here needs to be connected to the number 2 position. So that's where it's going to attack. When that attacks, too many bonds to carbon, so this one's going to pick up a proton. So that pair of electrons is associated with this carbon that picks up the proton, and now I have 2 protons here. I've made an oxygen bound to the second. The hydroxide on the 2 is bound, and I have fructose 6-phosphate. Okay? Now, one test question that I will never do is make you go all the way from here to here. Okay? One thing that I might ask you to do, I may not, would be going from maybe like here to there. Okay? And what I would give you is the beginning part and the end part and anything that is available except for a base and a proton, because you always have those available inside the enzyme. Okay? But if there's any other reactant, I will give it to you that you would need. All right. But we're not at that point yet. All right. Third reaction. What do I do? Inventory. So what is missing? Well, one, it's a kinase. Okay? It's a phosphofructokinase version 1. Uh-oh. If there's a version 1, what does that mean later? You're going to have a version 2? Yeah, so if you ever see that, that's a problem. Okay? In terms of you're going to have to distinguish the two. And the kinase is going to work on something that has a fructose that has a phosphate. Fructose 6-phosphate. Okay? And so I'm going to make fructose 1,6-bisphosphate. Okay? At the end. You got ADP is a diphosphate, right? But this is a bisphosphate. Both of them have 2 phosphates. What's the difference between a diphosphate and a bisphosphate? Someone just being willy-nilly? Okay?
Is it where the phosphates are?
Yeah. A diphosphate means the 2 phosphates are connected together. The bisphosphate says it has 2 phosphates, but they're not connected. They're on the molecule, just in different positions. All right. So this one kind of is helpful because you've got boxes, right? The hydroxide that is boxed loses a proton, right? And it gains a phosphate. Where does it gain a phosphate from? From ATP. And looking at the reaction, the oxygen, once it's deprotonated, will attack at the alpha, the beta, or the gamma? At the gamma. We've done 3 reactions. We've done our first repeat. This is just like the first one. The R is different. So here, a base. We'll take that proton. The pair of electrons that are between the oxygen and the hydrogen will then attack, oops, that's weird, attack the phosphate. What has to happen when I attack something that has a double bond? The electrons must what? Go up. And the electrons must come down. And what falls off is ADP, so this bond is broken and goes onto that oxygen. ADP is the leaving group. And I've got fructose 1,6-bisphosphate. Great. All right. This is an investment of ATP. The first reaction was an investment of ATP. I've used energy. Step number 4, enzyme called aldolase. This enzyme has more as a common name, and the other ones tell you kind of what is reacting and what goes on. But this one is a generalized name. It's a really old characterized enzyme. It's an ACE because it breaks down things, okay? And it's going to break a 6-carbon intermediate into 2 3-carbon intermediates. Essentially, you're going to break this bond right there, okay? And that's what we see, okay? And we get dihydroxyacetone and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Those are our acronyms. All right. Now, this reaction is done in a little bit different way. We always have a proton and we always have a base, but sometimes the enzyme actually covalently reacts with the intermediate as part of the reaction. It's a way to reduce the energy of a transition state. This is one of those enzymes that actually makes a covalent link and becomes part of the reaction. Okay? So I'm going to articulate this one a little bit more. Also, this reaction has to take place with an open ring structure. The first step in the reaction is the sugar is reacted with an amine in the enzyme. So E is the enzyme. And so the enzyme reacts at the ketone to create a hydroxide. Then the nitrogen, oh, that's backwards. The nitrogen is in the middle, gets deprotonated. It gets fixed in the third step to create that double bond, and the hydroxide leaves. So what happens is, instead of being a carbon double bonded to an oxygen, it's a carbon double bonded to a nitrogen that has a positive charge on it attached to the enzyme. The reason is this. When I take the base to break the bond, which the bond breakage occurs right here, okay, that breaks the two apart, all right? This, creating a ketone is good. This, going on to that carbon is really bad. A negative carbon is really bad. But I could kick it here, and then I can kick the lone pair onto the nitrogen. What happens is I go from a positive charge to a neutral charge. That is really low in energy, okay? If I just did it over here, if I took that base off here, I formed this double bond, I formed that, and I put the negative charge on the oxygen, that's high in energy, okay? I can either surround that by positive charges and lower its energy, but this enzyme doesn't do that. It lowers the energy by having a nitrogen and a positive charge, and when that reaction occurs, I get a neutral intermediate, okay? And that is great, okay? Now, one of the products I get are the one I want, okay? And so this is the glyceraldehyde 3-phosphate. But the problem is my other part is still stuck. Now, I said a negative charge on the carbon is bad, okay, particularly. And so there's an equilibrium. Now, the equilibrium is like 95% here, 5% there. This is the reactive species, but it's real high in energy. But once I have a negative charge, I can pick up a proton very easily, okay? And so as this reacts, it goes, and so the distribution keeps going, and I can actually make it go all the way. The last step in this, I will upload the mechanism on Moodle. I just don't have it there right this second. Is I'm still attached to the enzyme, and I don't want to be attached. I've got a double bond nitrogen. I really want a double bond oxygen. Well, to get a double bond oxygen, I need water to come in and attack, and the nitrogen will now have 2 protons. I have a hydroxide. It's really the reverse. A base will take the oxygen, create the double bond, and the enzyme leaves. I have my second intermediate, the dihydroxyacetone. Okay? And then I have my enzyme ready to do another round. And so this is a way to stabilize the transition state, is the enzyme becomes part of the reaction itself. I will upload that mechanism for you to see this afternoon. Okay. Oh, I said 95.5. It's actually 96.4. I was pretty close. Okay. And the equilibrium gets drawn over. All right. And we have now 2 pieces of the puzzle. All right. And class goes till, is it supposed to be 12:30? I don't want that. My other class starts at 1. I need to eat. Okay. I'm going to go ahead and stop the lecture here. But I want to pose a possibility. Can anybody in the class not make it here at 10 o'clock? Do you have something before? You have a class right before?
No, I can't make it.
You can?
Yeah.
Can you make it by 10? Or do you have another class that goes right up?
Yes.
Okay. All right. So what we're going to do is we're going to start at 10:15, but we're going to end at 12:15 every day because I need to get some lunch before my next class. And that class goes from 1 to 5, so that's a long day. So we'll stop here. What slide did I get? What number? Do you know? 35. Okay. I ended on 34, so the next one's 35. So we're right on track. We should be able to finish up next time. But I had some introductory things. Okay, so tonight I want you to watch those 3 videos that explain a couple things. There are going to be one about acids and bases and how they do, one about attacking ATP, just to reinforce that. And the last one is going to be about redox reactions, which we're going to see in the next couple reactions, so you understand what's going on with those. And then, but that's the only thing that I want you to do tonight, okay, is watch those 3 videos, okay? And so, if you subscribe to my channel, which I never thought I'd actually say, but you'll see when they pop up. You'll get a notification when I actually post the new one. So when I post tonight, I actually split them up in 2, so you'll get part 1 and part 2, but you'll see when they actually get posted up. And that's the only advantage of signing up. The only advantage for me is so I get bigger numbers. So, all right. Yes?
Do we need to get a textbook, or will these notes be?
Usually the notes are sufficient, okay? If people can understand the notes and describe the process from the notes, that works pretty well. If you don't understand either how I describe it or the notes, the book supplements it by explaining it in a different way. It's the same information, but somebody might explain it just a little bit differently, and that's what most people use. I will never take a question from the text and throw it out there. I take it from my notes. I can take every question and put it back to a slide in the notes. And so it's not something I will grab, a paragraph that I never covered. It'll be here. The book can explain it in a different way.